Hi there, Dr. Rehan Falari, and we'll be going through the histology of basic tissues. We'll be introducing through this class to the types of tissues, basic tissues in the body. So, and that's the whole point of histology, actually. So, generally, there are over 220 types of tissues in the body, and we refer to its group of cells as tissues. They have to be a group of highly specialized cells that are known to perform functions that benefit the organism as a whole. So those cells, before they are regarded as tissues, must be similar in structure. They must be similar as well in functions, and they must come from a similar embryonic origin. All of these have to be in place before we refer to them as tissues. So at the end of the day, when you look through the entire body, like we introduced in some of our basic classes, you would find out that all cells in the body, all the tissues in the body are one of four types. They are either epithelial tissues, or they are connective tissues, or they are muscular tissues, or they are nervous tissues. So the epithelial tissues generally serve the they serve the function of covering. They serve as covers, they serve as linings. Okay, while the connective tissues, they serve so many functions. And those can be summarized as supportive functions. Muscular tissues, as you know, serve to contract in order to aid your body movement, while nervous tissues serve the purpose of general body control. So, to that effect, these four main tissues, if you would like, MENC, that MENC could be a way for you to code them. So, organs contain several tissue types, and arrangement of tissues determine the structure and function of the organs. The way the tissues are arranged determines how the organ can function and what the eventual structure the organ is going to have. So, this is the whole point of histology. Is the whole point of histology. And like was indicated in our previous class, cells generally, before they form whatever they form, before they make up the tissues that they eventually become, they go through different phases. Cells generally go through phases in their, I mean, for them to divide, for them to form, and then divide. Just thought it's necessary to quickly mention this because it was part of what was meant to be uh, mentioned in a previous class. So the cells will go through mitotic division. So there is this reproductive phase and there is the resting phase known as the interphase generally. So during this phase of development, let me grab the pointer. So during this phase of the cycle of the cell cycle. This is where the division happens. So this is referred to as the mitotic phase, everything from here. While everything that lies between the end of one mitotic phase and the beginning of the next one is known as interphase. During this moment of mitotic division, that's when the actual division happens. The first stage of that is the prophase, when the cells, when the chromosomes are coming together, they condense, and then by metaphase, they align themselves. Uh, in the middle, in the center, you have the centrioles already taking their position at the opposite end. By anaphase, you see the sister chromatids. Those are the two versions of the chromosomes that have duplicated. You see them pulling apart in order for them to start arranging towards the new daughter cells that are being formed. So you see them pulling apart. That's ana. So they are separating themselves towards opposite end. By telophase, they have already ended up at the opposite ends. right? So we are now having two daughter cells, each of which is composed of a full complement of chromosomes and a full complement of fluid-like organelles. And then a stage that follows the telophase is known as cytokinesis, when the cells divide, when the daughter cells divide, and then you have one cell ending up giving two cells. So this is the mitotic phase, and this is how cells reproduce summarily. What you have on the other side of it, after this division has happened, then you start seeing the chromosomes within the nucleus entangle, they unbundle, they unwind, and then they become free, and then you start seeing the nuclear membrane, the membrane of the nucleus, you start seeing it fade away, degenerate, so that it allows for the chromosomes to fade out of the nucleus, and then you find them all around the chromosomal strands. You find them all around the cell, because at that phase they are not really dividing; they are only getting the necessary metabolic um, readiness for them to be able to start another mitotic division when the time comes. So interphase is generally divided into three phases. So the first is the G1, during which the centrioles replicate. The centrioles are these two organelles, that you have on the opposite side of the cell, and they are the ones that coordinate the distribution, the arrangement of the spindle fibers, such that they end up dividing, they end up pulling sister chromatids apart, and then uh, they become homologous chromosomes that get separated. So it's during the G1 phase that the centrioles get replicated. And then there is also an S phase after the G1 phase. And that's where you have the actual replication of the DNA material within within the cell. So the DNA material that will compose the chromosome, that is the actual time they get replicated. And after the S phase, which stands for synthetic phase. So usually G1 is coded as gap 1. So, and then S is known as the synthetic phase. Then the gap 2, that's the G2 phase. That's when you have the final growth and activity of the genetic materials, of the chromosomal materials before the actual mitosis. So this is a summary of the phases of the cell cycle. Going forward into our discussion about the cell, the tissues, right? It's also important to remind ourselves of what we said about the extracellular fluid or extracellular matrix. So this is usually a fluid, but can be more gel-like or calcified in cases of bones. So the extracellular fluid provides a medium for dissolving and mixing solutes. So it also provides a medium for transporting substances around the cell, whatever the cell has secreted. It's through the extracellular matrix that it gets transported. Then it provides a medium for carrying out chemical reactions as well. So going deeper into the epithelial tissue, now epithelium, so we have said that there are four categories of tissues, right? So the epithelial tissues is the first one that we're going to be assessing. And it's also known as the epithelium. And epithe means to lay something on, something that is laid on, or a covering, right? So that's why, that shows that epithelium refers to lining or covering, right? Uh, and some of these coverings get so sophisticated that they actually perform glandular. So it's from this epithelium that you have glands, some glandular tissues of the body forming. So it covers all the free body surfaces, and it contains versatile cells. So it's wherever the body has a surface, whether internal or external, you can be rest assured that the epithelium is playing a role there. So you have an epithelial tissue there. So nearly all substances that the body gives off or receives must pass through epithelium. Whatever is entering your body, entering your skin, is passing through the skin epithelium. Whatever is going through the blood vessels is going to be passing around the epithelium. Whatever sweat that comes from the body is passing through the epithelium, amongst other things. So the functions generally include protection, as with uh, any structure of the body that is encapsulating something, that is winding around something. So it offers protection, it offers filtration, such as helps to, filter fluids that are going out of the body so that the necessary nutrients are retained in the body before the wastes are exuded outside. It also helps with absorption, as with the gastrointestinal system, the alimentary canal, by which means you have the important nutrients that are within the food that have been taken get absorbed into the body. And you also have the case of secretion, as with glands. So epithelial tissue has these many functions. And among the characteristics is the fact that the cells fit closely together to form continuous sheets. So when they form this sheet, the sheet could be of single layer, the sheet could also be of multiple layers. And they usually have little extracellular materials, all right, because their job is to cover and to provide filtration, amongst other things. So the membranes always have one free surface or edge, okay, that is exposed to the body's exterior, or to the cavity of an internal organ. So some have modifications like cilia or microvilli. So when they are outside, when they are exposed, there is always a part of it that is closest to the surface. Okay, that's usually known as the apical surface. Whether that surface is directed towards the exterior of the body or directed to the interior of the body, in which case it will be the inner lining of a cavity. And when they are lying in the interior of a cavity, you can expect to find some of them having modifications like cilia, as we would see subsequently, the cilia that help to move substances along the route. So there is the membrane, right? We've talked about how those cells fit together. Then there is the membrane that is always having an apical part. So when there is an apical part, there is also always a basement membrane, right? So the epithelium is resting on the basement membrane, which is a structureless material. It's structureless because its composition, its configuration is always affected, is always indented, is always determined by the job that that epithelium is performing. We'll be seeing illustrative pictures shortly. So these structureless materials, that's the basement membrane, are secreted by both epithelial cells and connective tissues that border the epithelium. The epithelial tissues have no blood supply of their own, usually. So that's why we refer to them as being avascular. However, how do they get nourishment? How do they survive? They do survive because they depend on the diffusion of nutrients from the capillaries that lie in the underlying connective tissue for them to get food and oxygen living. So this epithelium, the epithelial tissues also regenerate easily. They regenerate quite easily. That is, they have a high mitotic rate. Many a times they get to shed off. Many a times know, they wear off and then they get replaced from time to time. They have nerve supply. And the funny thing is they derive from all the three primary germ layers. The three primary germ layers in embryology are the ectoderm, the endoderm, and the mesoderm. So you have the ectoderm as the most exterior, you have the endoderm as the most interior, and then in between them, you have the mesoderm. So being on the outside, the ectoderm generally ends up, it's a precursor to many linings of the body, both exterior, but most exterior linings of the body. So the skin, as well as a precursor to the nervous system. So it's from the skin. It forms the skin and the nervous system. The endoderm, which is most interior, uh, gives rise to organs of the gastrointestinal system. So organs, mucosae, glands, linings of cavities and tracts, while mesoderms generally result into the differentiate into connective tissues and most muscle tissues. So from these three, you will always find, you'll find these three in one way or the other contributing towards the formation uh, of epithelial tissues at different parts of the body. So looking at the classification of epithelium, how are they classified? Uh, so there are so they are classified either based on the number of layers, all right, because they are serving as lining. So if you classify them based on the number of layers, then an epithelium with just a layer will be referred to as simple epithelium, all right? While if it has more than a layer, we'll refer to that as stratified, like you can see here, having more than one layer. So another way of classifying the cells, the tissues, the epithelial tissues is by looking at the shape of that tissue, of the cells that make up that epithelium. So there are three main shapes they could have. When they are flat, they are referred to as squamous cells, okay? And the entire tissue will be referred to as a squamous epithelial tissue. While if it's not flat, if it's taller than being flat, if it's cuboidal, if it's like a cube where the height is similar to the width and length, so to speak, such cells are referred to as cuboidal as illustrated here. While they could also be tall, column-like, in which case we refer to such cells and such epithelium as being columnar epithelium. Therefore, there are two names that we use in describing epithelial tissue. The first one um, is going by that first classification we talked about, where it indicates the relative number of the cell layers, where we'll say simple, stratified, or when it's looking both single, if it's looking both simple and stratified, where it's kind of mixed up, such is referred to as pseudo-stratified. Pseudo-stratified, which means it is like being stratified. So it means it looks like having multiple layers. So these typically is a simple, this is a simple squamous layer, right? So this is a simple cuboidal, this is a simple columnar, right? So this is a stratified squamous, the stratified cuboidal, and this is pseudo stratified in the sense that it is one layer wedged uh, into the other. So the second name indicates the shape of the cells. So this is squamous, this is cuboidal, this is columnar, and then transitional kind of varies around. Okay, varies. So uh, so here we are, those same illustrations again, and the stratified epithelium is named based on the cell that you have at the topmost at the apical surface. Stratified epithelium is named based on cells at the free surface. Arrangement reflects location and the function of the tissue. So by seeing the arrangement of the tissue, you can tell what kind of a job that tissue is performing, and you can tell where that tissue is coming from in the body. So there you have again a pseudo-stratified columnar right here, stratified squamous. If it's stratified, you'll see some cuboidal beneath, but the most apical one, the one at the top, is squamous. That's why we refer to as refer to it as stratified squamous. Stratified cuboidal, pseudostratified, and then here is the transitional, where it varies what you have in the strata. Um, yeah. So the covering and lining epithelium will therefore be listed out as simple epithelia, in which case you have simple squamous, simple colloidal, simple columnar, and pseudostratified columnar. And when they are stratified, you could have stratified squamous, colloidal, stratified columnar, and then transitional epithelium. We can also have glandular epithelium. So there are things you need to know for you to be able to identify. So tissues, things you need to be able, things you need to be able to identify in tissues. So include all these things that we have just mentioned. And then when after looking at the tissue, so if you want to assess a tissue, like what kind of tissue is this? It means you want to be able to tell us whether it is simple squamous, simple cuboidal, simple columnar, and the rest. Now, when you look deeper into each tissue, what you wish to see further is the location of the nucleus of each cell, of the cells of that tissue. You want to identify where the apical, that is the free surface, is. You want to recognize the basal surface. You want to recognize the basal membrane. You want to observe if there are cilia there. You want to observe if there are goblet cells. You want to be able to recognize the keratin. So a simple squamous epithelium is a single layer and it has a flat shape. So all the cells in this case are attached to the basal membrane directly and they fit closely together. They form membranes where filtration or exchange of substance by rapid diffusion occurs. Things like absorption, secretion, or filtration occurs. You find simple squamous epithelium there. The examples are the air sacs of the lungs. We need such epithelium for respiration. So you find them also in the walls of capillaries for nutrients and gases to be able to pass between tissues, right? You need them in serous membranes. You find them in serous membrane. Those sleeky membranes that line the ventral body cavities and cover the organs in that cavity. So you find them in endothelium, which refer to the linings of blood vessels. So the linings of blood vessels are referred to as endothelium. That's the innermost layer of blood vessels, while the linings of body cavities and mesenteries are referred to as mesothelium. So all of them are epithelium, right? So mesothelium are those that line the body cavities and mesentery. So here are examples of these squamous cells, different example, different sites. Okay, so those are the flat cells you see forming a lumen around the air sacs, like in the lungs, similar body surfaces. You could also have simple cuboidal, in which case it's a single layer of cube-shaped cells. So here you see the common occurrence of this kind of epithelium in glands and their ducts, right? So examples are the salivary glands, the pancreas, walls of the kidney, tubules. They also cover all of ovaries. So here is a typical one. So these are taken from the lines of tubules, and here is a cuboidal cell. You will see that it's looking cube-shaped, finely arranged, forming this circle, leaving a lumen right within. So this is a simple cuboidal epithelial tissue. Okay, this is also a simple cuboidal epithelial tissue. So you can recognize here as the apical surface, which is the free surface that is exposed to that is directed to the lumen. This is the lumen again. So this is a cross section. That is, the structure is this long and then it was cut this way. That's what you have in this picture. While for this one, it's a longitudinal cut. That is, if the tube is like this, then you cut it halfway. That's what we refer to as longitudinal cut. So that's why you see it lined out this way. So the part of the cuboidal surface that is facing the lumen is the apical surface, while the base of it, okay, that's the basal surface that will be resting on the basement membrane. So that's simple cuboidal epithelium. You find them around glands, such as the salivary gland, pancreas. Then for simple columnar epithelium, so it's a single layer of columnar cells, which is like tall cells. So you often have this kind of cells, this kind of epithelium, often have goblet cells. Often have goblet cells which produce lubricating mucus. So you find them lining the entire length of digestive organs, digestive tracts, from the stomach down to the anus, right? Producing the lubricating with the goblet cells that are producing lubricating mucus. So when you find epithelium like this, it tells you that it's likely coming from the gastrointestinal tract. Okay, so here is a typical illustration of that. So here is the basement membrane, and I mean next to the basal surface of that columnar cell. You can see they are tall, and they are not cuboidal, and they are not flat. That is, they are not squamous. So here is the apical surface, and here is a typical goblet cell producing the lubricating mucus for that peristalsis. So as the digestion continues, the basal membrane for this one, the basal membranous surface for this one. So they often have goblet cells, and they often they can have mucosal covering. So they may be covered by mucosal membranes. So epithelial membranes that line the body cavities open to the body exterior. Epithelial membranes that line body cavities open to the body's exterior. So digestive tracts, like we mentioned, when it starts from the mouth, and digestion goes on as the food continues to the esophagus, the stomach, the duodenum, the jejunum, the ileum, the large intestine, the rectum, and then the anus. So it's going to communicate eventually to the exterior of the body. So after passing through those mucosae or mucosal membrane. So like we said earlier, pseudostratified columnar epithelium, so they appear to be multi-layered, so but are actually one single layer that rests on the basement membrane. So the nuclei of those cells, you will notice that in this previous, in all these illustrations, these are the nucleus, very darkly stained, right? So those are the nuclei that you try to identify. The rest of the component is cytoplasm with other organelles there. But this is using a stain that concentrates on delineating, letting you recognize the nucleus different from the rest of the cytoplasm. So here, the nuclei always appear at different heights. You'll see that the nuclei for these previous ones are seemingly rhyming, they're synchronized, okay? They're synchronized, both for the columnar, right? So both for the columnar and the cuboidal that we showed in the previous slide. So for the pseudostratified, they are not that arranged. They appear at different heights, and some cells are shorter than others. So mainly functions in the absorption and secretion. That's the main function of the pseudostratified columnar epithelium. So they can be ciliated, that is, they have cilia. So in which case we refer to them as pseudostratified ciliated columnar epithelium, right? So they also can have goblet cells, and you find examples of these in the respiratory tract. So with mucus being produced by the goblet cells in this epithelium, trapping dust and other debris. So this you can find in the respiratory tract. You'll also find the cilia propelling the mucus upward and away from the lungs. So that's what you have here. So here are cilia. So you can see the cells are of different heights. The nucleus are of different heights. They are not rhyming. So it's a single cell, but it's pseudo-stratified. It's not stratified, but pseudo-stratified. It looks like being stratified. What makes it look like being stratified is the fact that some cells are shorter than others than the other. And that makes their nucleus also not placed at the same height. You see the goblet cell here, and you see the cilia showing there. These are pictures from the respiratory tract, lung tissue, and others. Stratified squamous epithelium. So that means the cell has multiple layers and is flat. So they are most common stratified kind of epithelium in the body. So they are the most common stratified epithelium in the body. The cells at free edge are squamous, and those close to the basement can be cuboidal or columnar. Like I mentioned earlier, you name it stratified squamous or stratified cuboidal or whatever, based on what kind of cell is at the apical side. So they are found in sites that receive a good deal of abuse or friction. That's the stratified squamous epithelium. So keratin is a protein coating that you find on the apical surfaces of such stratified squamous epithelia, and they can be keratinized or non-keratinized. You find such in the esophagus, and of course, the ones in the esophagus and the mouth will not be keratinized, while the ones you have on the outer portion of of the skin will be keratinized. So here are examples, keratical illustration. So that's the keratin, and here is the apical surface of this epithelium. So here it does not have keratin, so it's non-keratinized. And here is the basal. So it is stratified squamous. All the cells are flat, and it is stratified up onward. While this one, all the cells are, yeah, they are stratified. You see the squamous are the most populated towards the apical region for both of them. And so this is the keratin, and here here is the getting close to the basement membrane, basal surfaces of the cells. And you could have the stratified cuboidal epithelium, where two cell layers with at least the surface cell being cuboidal in shape. So fairly rare in the body, and the distribution is extremely limited. So you mainly find them in ducts of large glands, with larger ducts of mammary glands, sweat, and salivary glands, as pancreas. So major secretory organs of the body. So it's quite rare that the stratified cuboidal epithelium, and here is the example. So yeah, and next is the stratified columnar epithelium, which, as the name suggests, stratified columnar epithelium. So it has multiple layers of columnar cells, and the basal cells vary in size and shape. And they have so therefore giving multiple distribution of the nucleus. Even less common in the body, uh, and the distribution is extremely limited to so much less common than even the stratified squamous that we mentioned earlier. So they are mainly found in ducts of large glands too, and you find them in the urethra, the stratified columnar, and you find them in the pharynx. Well, so transitional epithelium has variable shapes and it's highly modified, such that it's highly modified with stratified squamous epithelium that forms the lining of only a few organs. And such includes urinary bladder, ureters, and part of the urethra. So the transitional epithelium is so structured in line with its function of being exposed to considerable amount of stretching, right? So um, the cells of the basal layer can be cuboidal or columnar. So like we said, yeah, so they can be cuboidal or columnar. And the cells at the free surface also vary in appearance. So it's really transitional. So they vary in its appearance and arrangement. It's usually not stretched, and the superficial cells are rounded and dome-like whenever it's not stretched. Whenever it's not stretched, the superficial cells are rounded and dome-like. Or whenever it is stretched or distended, the epithelium thins out, okay, and the surface cells flatten and become squamous-like. And this is what we are seeing here in this urinary tract. So you can see this distribution of the cell is quite transitional. You can see some cuboidal towards the base. Yeah, and then you could see the cells or the center, I mean, at the apical surface. So when it is not distended, when it is at ease, so the cells uh, look, they look at ease, and then you see the superficial cells being rounded. While in the distended state, you see that the cells are beginning to flatten out. So there are also glandular epithelium. So like I alluded to earlier, the gland, you have one or more cells that make and secrete a particular product. You have one or more cell in the gland secreting a particular product. So that's what we refer to histologically as a gland. So secretion typically will always contain protein molecules in aqueous fluid. And for endocrine glands, they are usually ductless glands in the sense that they secrete their contents um, directly into the bloodstream. They diffuse directly into the blood vessels that move through the glands. So they are endocrine, right? An example of such glands are thyroid glands, the adrenal, and the pituitary. While for exocrine glands, they have ducts. They have ducts. So because their contents are not being emptied directly into the bloodstream, usually their secretions are emptied into the external environment, and the secretions empty through ducts. That's why they should have ducts. So they empty through ducts to the epithelial surfaces. And this is what the case is with glands like the sweat gland, the sebaceous gland, oil gland. That's the case with the liver, with with the pancreas. So here is typically a micrograph of the colon. So generally, this is a chart that helps you to remember what each of these um, epithelium stands for, where they are found, what functions do they perform. So you can go through them for better familiarization based on what we have discussed. So the transitional epithelium is known to line the bladder, the urethra, and the ureters. It allows the urinary organs to expand. Stretch. So speaking of the membranes, this chart I put it there for you to just further appreciate how diverse the kind of membranes that this epithelium lie could be. So here you can see mucus secretion right above the cilia here, and then this is the actual epithelium. So you see the basement basal surface, the basement membrane, under which you have the lamina propria in the case of areolar tissue. So here is the mucous membrane. As for serous membrane, you have the transudase right on the top. So those cilia here. And then the lining, the mesothelium, and then the areolar tissue. While for this cutaneous membrane, you simply have an epithelium of different types. And then you have the areolar tissue, you have dense irregular tissue. These are the different kind of ways that the membrane could appear. So from this point, so we'll move straight into the nervous tissue. Okay? Move straight into the nervous tissue. So it serves the purpose of internal communication. So that was us summarizing what happens in the epithelial tissues, right? So the next we're considering is the nervous tissue. It serves the overall control of the body. And the nervous tissues are very important for internal communication of one part of the body or one system of the body to another. So it's found in the brain, the spinal cord, and in the nerves. So those are the nervous tissues, whether it's the central nervous system or the peripheral nervous system. So two categories of cells occur in the nervous tissue. Some of them, the first category of them are the ones that do the actual conduction of the electrochemical impulses from one part of the body to another. That's referred to as the neurons. The neurons, those are the first category of cells in the nervous system, while the other category of cells work to support the neurons, and those are what we refer to as the neuroglia. Neuroglia. So they are the supporting cells that insulate, the support, and protect the delicate neurons within the structures of the nervous system. Okay? So the neurons receive and conduct electrochemical impulses, and they have two functional characteristics, and one that is irritability and then conductivity, right? So that's the neurons. They have a cytoplasm that is drawn out into long processes. So the long process is the longer one is usually the axon. Then you have dendrites branching off. So here is the axon extending long this way. Then the dendrites are coming up here. So before I say that, I should have said that the neurons, a typical neuron, is made up of let's say three main parts for the sake of simplicity. So one is the cell body. That's where you have the structure, the characteristics of a typical cell. So that's the cell body of the neuron. Okay, that's where you find the nucleus and all other organelles of that cell, right? Then you have the dendrites. Those are the processes that receive impulse. Those are the processes that receive impulse. While after the impulse has been received and then an action potential has been generated as a region referred to as the axonal hillock within the cell, within the cytoplasm, then those impulses um, get processed and sent out through the axon. All right. So here are typical illustrations of the neurons. While for neuroglia, they are supporting cells of the nervous tissue. They don't generate nerve impulse. Okay. Rather, they perform various supporting roles depending on what type it is we are referring to. They are often the site of tumors because they can divide, often the site of tumors because they can divide very fast. And then they insulate, support, and protect the neuron. So there are 6 or 7 categories of them. So you find some of them in the nervous system, I mean, in the central nervous system, find others of them in the peripheral nervous system. I can spare you of those details for now. So these are our concluding slides. These are our concluding slides, which we can elaborate on further. So the concept of tissue repair, wound healing, amongst others. So we'll revisit this after other uh, types of issues have been reviewed. Okay, the concept of inflammation and the concept of granulation, and then regeneration and fibrosis, those factors that affect tissue repair. So all of this we would address afterwards so that it makes much more sense. So at this point, we will be pausing. I'll be happy to take your questions. So, so I wish you all the very best and see you in the very next class.